virtual meeting has opened the meeting to those who haven't been able to join in the past at the Smithsonian. So that's been a big benefit for us, and I think allowed a lot of people the opportunity to sit in on these calls and listen to our presentations. Gary, are you online? He may be having a little trouble getting started. All right, we'll check back with him in a few minutes and then have him read through the meeting minutes from last meeting. Christina, can we move forward with you? Can you provide us with a reading of the new members?
I can provide you with a partial reading of the new members. Thanks to the fact that everything has gone virtual, I don't think I realized until I was compiling the list how packed my inbox had become. So if you are a new member and I neglect your name, because I only have 4 names and I know for a fact I input more into the database than that. So we have Yuda Nagano, Roy Smith, Francisco Infante Martinez, and Imelda Guadalupe. I know there's more of you. I'm so sorry that I'm struggling to find all the emails. I think after a certain point, I started deleting extra emails I had. So if I deleted your membership, I'm not deleted you should still be a member. You should still be a member. If I deleted that initial email of me knowing you're become a member, my apologies. I will do better in the future. Things I think are still, well, I'll do better in the future and then someone else will take over. So hopefully they'll do a better job than me. Regardless, those are our current members that I have a list for. If any other new members would like to speak up, that's perfectly fine. If you'd just rather stay quiet, that's also fine with me. I will stop talking now.
Thank you, Christina. Let's check back to see if Gary is on the line. Gary Hebel. All right. He did talk to me today. Did you get a chance to talk to you, Cece, to see if, okay. Well, I don't know what the issue is there. All right, so let's move forward to new business. Anybody have new business that they'd like to bring to the meeting today? All right, I have on here Young Entomologist report. Do you have anything to add to this, Dave?
No, I, as you know, finished the report for this year, and I just decided that the Young Entomologist group remain dormant until next year, where we'll have a full slate of activities, whether they be virtual or outdoors. It's going to be hard to say at this time. Yesterday was the largest national number of cases for the virus. We topped over 100,000, and I know in the area it's been spiking in Northern Virginia, Maryland, and even in the district. So I just have a feeling it's going to be a while before we can open up like normal. So we're going to start virtual, and you know, we've been shut down all year using parks and places that we normally go outside. We're not allowed to use them when we have groups over 5. So we'll start in February.
Okay. Well, thank you very much for that update, and we look forward to everyone starting back up, even if it is virtual. It's a wonderful program. I do want to say, as part of business, it's not really new, but regular business, we are ending the year 2020 for the ESW. So final reports will be gathered and presented in our next meeting, and you know, we'll present that, and then this will change over to the incoming president, Jamie Zonizer, in January, well, end of December. So, any other new business that you'd like to bring forward, anyone? All right. Do we have a virtual count of those on the call today?
It looks like 18.
Okay, well, that's good.
Cheryl, would new business include officer reports?
Of course, you could.
Okay, I just wanted to say hello to everybody. If I haven't, if I don't know you or I haven't met you, my name is Jamie Zonizer. I'll be the president next year, currently president-elect. And I just want to let you know that this meeting is usually, the November meeting is usually the one where we would announce the nominees for officer positions for next year. And so we have a nominating committee together, and we have people in slots for most of the positions, but we're still waiting to hear back on one. So we thought we would wait till we have the full list that we can email to everybody. And we'll of course do that before the December meeting, which is where the elections take place. And if you haven't seen them before, it's not very controversial. There's usually just one nominee per position, and it looks to be the same this year. But yeah, so I just want to say it's coming together, and you should see an email about it when we have all the nominees together. Thanks.
Thank you, Jamie. All right, virtual count, we got presentation
19 right now.
Oh, good.
Sorry.
No, that's fine. Have you seen Gary join yet? Okay.
Not yet.
All right. And we are recording, correct?
Correct.
Okay. Okay, good. That way we'll have some minutes to uncode. All right. Do we have any presentations of notes or exhibitions, new specimens, books? Okay. I would like to introduce Cece to do the presentation of the speaker, and then we will have a nice presentation. We'll get back to you after that is over.
I will actually be handing things off to Dave, as Dave is the mentor of Ian, who will be presenting from Young Entomologists.
Super.
Thanks,
Also,
Cheryl.
I will be muting everybody, that way only Ian is speaking.
Thanks, Cheryl and Cece. Actually, just quite unusual. I've known Ian before he was born. When I was working at the museum many years ago, his mom, Marion, was my volunteer, and she was carrying Ian. So I actually knew a lot about what was happening and how he was moving and things like that. And there seemed to be a little hiatus after he was born, but every now and then Marion would let me know how things were going. And it wasn't until, I think, 2012 when the Young Entomologist started officially as being a outreach program for ESW, where I met Marion and Ian again, and they were one of our original 6 families that started the Young Entomologist group. So I would see them quite often. Ian was always interested in environmental science and nature, and had always collected the most unusual insects in the area. And so we developed a relationship where some years ago, actually 3 years ago, he'll talk more about that in his presentation, where he actually decided to work with me on a study that actually was put into my ear by Gary Miller. Gary is the aphidologist at the Beltsville Agriculture Research Center, Systematic Entomology Lab, part of USDA. And I worked for him for a short time, and he got me to looking for aphids that I might find on Jack-in-the-pulpit. And I was able to find some, and then with Ian's interest, I got him involved more broadly, not just looking for aphids, but for other insects and arthropods that might have some association or some by chance association with mature forest where these plants live. So with that, well, I can tell you more. Don't fall asleep. So Ian is now a senior. He is in school as a student at Frederick Community College. He's also a homeschooler. And tell you the truth, I didn't know anything about homeschool until I met some great students, and Ian being one of those. And I have the utmost respect for homeschool education now. Something I never really understood or had any privy to until I started to meet students homeschooled through the Young Entomologists group. So I'm very impressed with them, and I'm very impressed with Ian, and I think that you'll feel the same after this presentation. So without any further ado, I'll introduce Emanuel.
Thank you for that very nice introduction, Dr. Adamski. And, so as Dr. Adamski just said, my name is Emanuel. He really just fully introduced me there, and I don't really have much else to say. So I think I'll just start with introducing my project. And as Dr. Adamski said, my name is Emanuel. And this presentation is on the 3-year internship I have had with Dr. Adamski, focusing on insects associated with Jack-in-the-pulpit. The purpose of this project is to find insect-plant interrelationships for the Jack-in-the-pulpit plant. Several studies have been done studying insect relationships with Jack-in-the-pulpit, but most of the results proved inconclusive except for a few very key relationships found with the Jack-in-the-pulpit plant, which I will talk more about later. There were also undescribed species of insects observed on the plant. As Dr. Adamski mentioned in my intro, I think he mentioned that there were undescribed species of aphids observed on it. And there were 2 undescribed species of aphids collected earlier this year. This research will also help us gain a greater understanding of the mature forest ecosystem, because Jack-in-the-pulpit is actually strongly associated with mature forest ecosystems, and not much is known about them in the first place. Jack-in-the-pulpit is in the arum family, which has more than 3,500 species distributed among 100 genera worldwide. Only 5 native genera are found in the northeastern United States, and this is represented by just a minority of the species. Arums are found on every continent except in the polar regions, and all species have a spathe and spadix. And here on the right, there is an image of the Titan arum, the largest flowering arum in the world. And when people first look at the Titan arum, they think this whole plant is the flower. Yeah, that's what it really looks like, doesn't it? Well, actually, what this big singular petal looking thing is a spathe. A spathe is a modified leaf that wraps around this fleshy spike, also known as the spadix. The spadix tends to have smaller flowers attached to it. And in this case, the spadix has flowers attached at the base, which are hidden by the spathe. It's very interesting because many people will think that this whole structure is the flower, when in reality, the flowers are hidden from view by the spathe. Many members of this family produce an odor resembling decaying flesh or decaying organic matter, and this attracts pollinators that feed on carrion. And there are also, many of these species contain calcium oxalate crystals. They are very irritating to consume, and if consumed, can cause damage to the vocal cords. Jack-in-the-pulpit is my study. Jack-in-the-pulpit, or Arisaema triphyllum, is my study plant here, and it grows mostly around wetlands, but also in places that have rich, moist soils. Its geographic location is central to eastern United States, and there are also some populations that have been documented in southern Canada. And it also grows in mature forests, as I mentioned earlier. The map on the right shows the distribution of Jack-in-the-pulpit in the United States. And the image on the right, or I'm sorry, that was the image on the left. The image on the right is of my collection site, Mount Briar. This image was taken in my first year of collection in 2018 in very early spring. And it showcases just very good habitat. And the only thing that is up in the background are the skunk cabbage in the back, which leaf out very early. As I mentioned earlier, Jack-in-the-pulpit is a member of the arum family, Araceae. It is herbaceous and dioecious, which herbaceous means that it dies back every year, and dioecious means that it is functionally either male or female. Even if the plant has both male and female flowers, only one sex will function properly, making it a truly dioecious plant. Like all other members of the arum family, Jack-in-the-pulpit has a spathe, which wraps around the spadix, poking out, and the spadix has flowers at the base of it. Jack-in-the-pulpit also has 1 to 2 leaves, each of these leaves having 3 leaflets each. Its fruit is a cluster of green berries that turn red as they mature, and it also has a corm, which differs from the root system as it serves as a food storage area. Here's an image of, or here's a line drawing of a Jack-in-the-pulpit with the leaf with 3 leaflets, the spathe, which wraps around the spadix, and this spathe is illustrated to have male flowers at the bottom, the fruiting body, and the corm and stem. Last slide, I actually mentioned that Jack-in-the-pulpit plants can have 1 to 2 leaves, and this one is only illustrated with 1 leaf. The reason why it's only illustrated with 1 leaf is because it's depicted as a male plant. And male plants are generally smaller than the female plants male plants are generally smaller than female plants. And this means that male plants usually only have 1 leaf, while female plants will generally have 2. Here's an image of a freshly pulled Jack-in-the-pulpit. You can see the corm with the root system coming out above it, and then next year's sprout coming out of the corm. And here are photos of inflorescence. On the left is an image of just a spathe. In the middle is an image of a female plant spathe that has been cut away to reveal the female flowers. And on the right, there's an image of a male plant spathe that has been cut away to reveal the male flowers. What's really interesting about the female flowers is that even though they look like unripe berries, those are really just enlarged ovaries. And interestingly enough, there are actually differences between the male plant spathe and the female plant spathe. The male plant spathe has this opening at the bottom of it, while, as you can see in the female plant spathe, lacks it. It is theorized that this opening is to encourage pollination between the plants, because if an insect flies into the Jack-in-the-pulpit spathe, it will not be able to get out the way it came in, due to the fact that the spathe walls are very slippery and they cannot crawl up them. So they would go around the bottom of the male plant spathe, picking up loose grains of pollen on their bodies, and hopefully they would eventually find this opening. which they would crawl out of and potentially go to another spathe. Let's say the next spathe they visit would be a female plant spathe. And as you can see, the female plant spathe has no hole at the bottom, so they would fly in and they would not be able to get out. They would just go around distributing the pollen grains they have on their body from the male plant spathe among the female flowers, and they would effectively pollinate it. However, the insect, once it goes in, unless it has a method to get out of the spathe, it will die in there. Jack-in-the-pulpit is a perennial plant, which means it lives for over 2 years. It has 2 methods of reproduction. It can reproduce asexually by cloning itself, in which it shoots off a little cormlet, and this cormlet acts as a new sprout. However, the cormlets are genetically the same as the parent plant. And it can reproduce sexually by cross-pollination with a plant that is genetically different than itself. Jack-in-the-pulpit is very interesting because it actually changes gender, or it can change gender within its lifespan. It starts off as a sprout with no inflorescence, and as it's able to grow bigger and its corm is able to store more carbohydrates, it can produce its first inflorescence, which will always be male. This is because the female reproductive parts require much more energy than the plant has available to produce, or the plant has stored. So as the plant gets bigger and is able to store more carbohydrates in its corm, it will eventually be able to produce a female flower, and by which the plant will be female. Jack-in-the-pulpit can only change its gender once a year, and it's theorized that it changes its gender from sometime in the fall to winter before next year's sprout has come up. And what makes Jack-in-the-pulpit unique among arums is that a female plant can actually revert back to a male plant, and a male plant can revert back to having no inflorescence. Members of the arum family can only change gender in one direction. A male plant could become a female plant and it would never change back, or a female plant could become a male plant and never change back. I collect around April through June each year, and due to the heavy rainfall event we had in 2018, I did not collect much after May that year. I did, however, make observations after them. The bloom time is around April through June, and the blooms last from 2 to 5 weeks. However, since all the Jack-in-the-pulpits don't come up at once, the collection period is extended for more than those 2 to 5 weeks. Here is a map of my 4 initial collection sites. 2 of them were located in the northwest part of Maryland, more northwestern Maryland, and 2 of them were located more in central Maryland. After my first year of collection, I dropped 3 of these sites due to logistical reasons and continued collecting at Mount Briar for the rest of my collection seasons. Here's an image of Cunningham Falls State Park. This was taken in 2018, my first collection year, and is a good example of a forested wetland. Here in the foreground, you can see these ferns are actually cinnamon ferns, which are known wetland plants. In the middle of the image, there's more skunk cabbage. Skunk cabbage is actually in the same family as Jack-in-the-pulpit. It is an arum, but skunk cabbage actually blooms at very different times and leaves out at very different times than Jack-in-the-pulpit. The skunk cabbage blooms in winter and can even bloom when there's still snow on the ground. And what's really cool about it is if there's snow on the ground and it shoots its flower up, or it shoots its spathe up with the flower, the spathe produces so much, or the spathe can produce so much heat that it melts the surrounding snow. And it produces a foul odor like most other air species of the arum family, and which attract many pollinators, and which mainly include flies and carrion beetles. Here on the left, there's a pawpaw sapling right here. Pawpaw can grow in either wet or dry habitats, so it's not really a wetland plant. And the site is just very lush and it's very wooded in the background. So it's just a great example of where you'd find Jack-in-the-pulpit growing. Here's an image of Treville. This is in Rockville, also my first year of collection. And you can see there's a kind of a gradual slope over by the development leading into this depression housing the skunk cabbage. Wetlands wetlands usually form at the bottom of these gradual slopes or hillsides due to the fact that the water will tend to pool there, and then the water will sit there, and plants that love water will grow there. These areas, these kind of depressions retain water very well, and as you can see by the skunk cabbage, the flared roots on this tree, it's definitely a wetland. Interestingly enough, Jack-in-the-pulpit is usually found on the outskirts of these skunk cabbage populations. And that is because Jack-in-the-pulpits don't really like the areas that are completely inundated with water. Instead, they prefer the areas, the drier parts of wetlands, if that makes sense. And here is an image of Beltsville. I found this basketball and Dr. Adamski in the woods. There's a lot of skunk cabbage just growing around. On the right here, there's a spice bush sapling. It's a little bit hard to see. And in the back left, you can see maples blooming. Spring is really coming in here. And as you can see by the basketball, the tire in the bottom right, and the scrap of cloth on the tree over there, this place has been impacted by human land use and human use in general. There is a pretty busy road right next to the site, and there's a lot of trash littered around, degraded areas of habitat, and it has been impacted by human land use. Another very interesting thing about this site is that it is in the coastal plains geographic area, and coastal plains is generally known as being very flat. And as you can see by the from the site, is very flat. So that's just an interesting correlation. And here is the same image from Mount Briar in very early spring before anything was really up. And it just shows very good habitat. There are still a few things up, however, like the skunk cabbage colony in the background and the blooming serviceberry. There's a mud patch that is particularly wet right there. There's clumps of sphagnum moss around, and this tree even has slightly flared roots. The main trees you will find in this kind of habitat would be maples, ash trees, slippery elms, and box elders. Here's another image from Mount Briar. This was actually from my first collection date of this year. And it's a little bit later in the spring than the last image, but it's still pretty early in spring. But you can see there's sedges and grass just coming up around. I'm collecting from a Jack-in-the-pulpit plant right there. And as you can see by my hat and rain jacket, it was very cold that day. Here's an image of Jack-in-the-pulpits that are just coming up. This was taken 3 days later than the last picture on April 19th. And Jack-in-the-pulpit, when it first comes up, will always shoot its spathe up first, as you can see right here. And then the leaves will follow. And you can tell a Jack-in-the-pulpit has just come up because its leaves look very droopy and generally ill. However, they will settle out eventually and look more flattened and uniform, like this Jack-in-the-pulpit plant on the left. These were also taken on April 19th in Mount Briar. The image on the left shows me collecting from a Jack-in-the-pulpit spathe. The image in the middle is of a Jack-in-the-pulpit that has most likely recently come up due to the time of the season. And the image on the right is of a Jack-in-the-pulpit that has definitely come up very recently. As you can see from the leaves have not yet settled out, and they still look kind of droopy and ill. Here's an image from May 2nd, and spring is now in full force here. As you can see, the sedges and grasses have taken over the ground cover, taken over as a ground cover. There's very small patches of jewelweed around, and the canopy has leafed out. The image on the left is actually the same skunk cabbage colony that was in the image from 2 years ago during my first collection season. And same thing with the Treville site. It's at the skunk cabbage colony is at the base of a sloped hill, and it is formed in a depression where water will pool. And the image on the right is of a serviceberry in fruiting. This image was also taken on May 2nd of this year, and this is of the central part of Mount Briar. Now, the central part of Mount Briar, as you can see, is inundated with water, and not much will grow in the parts that really have a lot of water, except for grasses will grow on the shallower parts and the raised parts in the water. You will mostly find Jack-in-the-pulpit growing along the drier strips, like the one in the middle of the photo. So my general procedure for collecting was first I would sneak up on the plant and observe the outside. This is because I was in part looking for undescribed species of aphids that were known to be on the outside of this plant, and also any other unique insects or arthropods that I would observe on the outside of the plant, I would collect. After checking the outside of the plant thoroughly, anything of interest I would see, I would collect with a brush dipped in either alcohol or water. For the most part, I would try to use water because I was collecting from the same plants a lot, and especially when collecting from the Spathe, because water won't really leave behind that alcohol scent, and the alcohol I feared might disrupt the insects from visiting the Spathe. But for some of the bigger specimens, I did have to use alcohol to kill them or knock them out and get them out of the Spathe. After dipping my brush, I would then sweep the brush around inside of the Spathe. Even if I did not see anything inside the Spathe, I would still sweep around anyways, because sometimes there were some really small things that I only got because I swept around inside the Spathe. After collecting anything inside the Spathe, or even if I didn't see anything, I would dip my brush in the vial of alcohol. I would then label vials with locale, date, and collector, any other important information after my collection day. And after the collection season, I identified specimens to family with Dr. Dempsey's help. Certain specimens were ID'd to species when needed. This includes the specialist thrips, Heterothrips Arisaema, that were identified by Dr. Cheryl O'Donnell. The image on the left is of Dr. Adamski showing me how to collect during my first year. And the image on my right, or the image on the right, shows a crane fly on the outside of the Jack-in-the-pulpit Spathe. This is just an example of why you always check the outside of the plant before collecting. It's a very obvious example that you probably wouldn't miss if you looked at it, but it just shows that sometimes you'll find something very unexpected on the plant, and if you disturb the Spathe before looking at the rest of the plant, you could lose the opportunity to collect something very unique. And these are images of arthropod families that I collected over the past 3 years. They're ordered from most numerous collected to least numerous collected. And so we start off with Mycetophilidae and Cyaridae, the fungus gnats and darkwing fungus gnats. These two families have previously been associated with Jack-in-the-pulpit from previous studies. And these, along with another family that will be mentioned here in a second, have been claimed to be the main pollinator of Jack-in-the-pulpit due to the sheer amount of numbers of bees found in the Spathes in other studies. We then have Psychodidae, the drainflies, Chironomids, the midges, Ceratopagonidae and Blepharisaurids, the biting midges and netwing midges, and then we have Thripidae, the thrips. These thrips are the specialist thrips that use Jack-in-the-pulpit as their host plant, Heterothrips Arisaema. And these guys, like I just said, use Jack-in-the-pulpit as their host plant and feed on different parts of Jack-in-the-pulpit in different life stages. And these guys are what many of the previous studies stated were the main pollinator of Jack-in-the-pulpit. This is most likely due to the sheer numbers, because the colonies inhabiting Jack-in-the-pulpits are very large, but they cannot go more than a year at a time. While fungus gnats can fly a lot better, but they are a lot less numerous than thrips. And here we have Alirotidae, the white flies, Phoridae, the humpback flies, Cecidomyidae, the gall gnats, Dolichopodidae, the long-legged flies, Topulidae, the crane flies, Cicadelidae, the leaf hoppers, Empididae, the dance flies, and finally I collected 2 different families of Collembola, Entomobriidae and Sminthuridae, the slender springtails and the globular springtails. These guys are examples of why I swept around inside the Spathe even though I didn't see anything, because I definitely did not see these guys, and especially with the low visibility inside of the Spathe, this was only collected because I just swept around. And now for aphid information. There were aphids found on both the stem and the corm, and there were 2 undescribed species found, which were Rhopalociphem species and Pemphigus species. There was 1 described species of aphid found on the stem of the Jack-in-the-pulpit, which was Mysis persicae. However, Mysis persicae has not, to my knowledge, has not been observed on Jack-in-the-pulpit previously. And the aphids were identified by Dr. Gary Miller of the Beltsville Agriculture Research Center. The image on the left is of the winged form Mysis persicae, and the image on the right is the winged form of the Rhopalociphem species. And on the left, there's the wingless form of the Rhopalociphem species. If you've ever seen A Bug's Life, the queen ant has a pet aphid that looks a lot like that, and I just thought that was pretty cool. And on the right, there's the wingless form of the Pemphigus species. Here's an image of the Pemphigus species on a pulled up Jack-in-the-pulpit. This Jack-in-the-pulpit was pulled up at Little Bennett State Park. And the Pemphigus species are a little bit hard to see, but there are these little round things at the base of the stem nearing the top of the corm. And on the right, there's a zoomed in image of the Pemphigus aphids, and some of them are even secreting wax blooms. I also collected some predators. The main predators I collected were spiders and hemiptera. And in my first year of collection, there was an immature assassin bug on the hood of the Spathe. And there were also a lot of spiders collected as well. And there were many different spiders that used the Jack-in-the-pulpit parts in many different ways. There were some spiders that made webs in the hood of the Spathe as directed by the arrow. Some spiders would just sit in the base of the Spathe waiting for their prey to come in. Some spiders would make a very messy web in the base of the Spathe to catch anything come flying in. And some spiders would use the small space between the 3 leaflets of the plant to make a very fine web. And here the image on the left is of a spider sitting on the spadix waiting for its next meal to arrive. And the image on the right is of a small spider trying to drag a much larger fly across the leaflets. And here is my data comparison for 2018 through 2020, which is this year. 2018, or data from 2018 is in green, data from 2019 is in orange, and data from this year is in blue. And the most numerous 6 families collected were Mycetophilidae and Cyaridae, the fungus gnats, Psychotidae, the drainflies, Ceratopagonidae, the biting midges, Blepharisauridae, the netwing midges, and Chironomids, the regular midges. The interesting thing about these families is that all of them are in the suborder Nematocera, or the primitive flies. If you go down the list a little bit, you'll have Thripidae, Dolichopodidae, and Alirotidae. Thripidae is Thyssanoptera, Dolichopodidae are in the suborder Brachycera, and Alirodidae are hemipterans. And if you go down the list even further, you'll see that there's a very wide variety of insects. I only collected 1 to 2 species, or 1 to 2 specimens of each family. These interesting families include Scolididae, the bark beetles, Bibionids, the love bugs. They're called love bugs because whenever they are seen, it's usually when they're mating. Cycadidae. There was a bagworm that attached its bag on the inside of the spathe. Like most of these other 1 and 2 off families, it's probably by chance, but it's still very interesting nonetheless. Tetrygid, the pygmy grasshoppers. Braconidae, the parasitic wasps, or a family of parasitic wasps. Lithriidae, which these guys are actually known as fungus beetles. And it's very interesting because Jack-in-the-pulpit emits a slight fungal odor, and there could be an association there, but only 1 of these beetles were collected over the past 3 years. Psyllidae, plant lice. The Collembola, Entomobreedidae, and Sminthuridae. Nabidae and Rediviidae, which both families are known to be usually predaceous. And finally, Buprestidae, which are the metallic wood-boring beetles, the most notorious member of this family being the emerald ash borer. And if you look at the graph, it shows that this year's collection had a lot more in numbers than 2019, and an even greater difference in 2018's data to this year's collection data. This can be attributed partially to the fact that I collected only twice in 2018, 5 times in 2019, and 8 times this year. 2018, I was not able to collect much due to the extended rainfall event, and each year I collected 3 more times than the last year. However, there were some families like chironomids and cyarids. Both had very similar numbers collected over the 3 years, which is very interesting due to the differences in collecting dates. And finally, the thrips collected only numbered to a total of 13, while Mycetophilidae and Cyaridae were much higher in numbers. With many studies saying that thrips were the main pollinator and the minority of studies saying that Mycetophilidae and Cyaridae were most likely the main pollinators of Jack-in-the-pulpit, I think at least in Mount Briar, thrips are definitely not the main pollinator. And it's more likely that not only Mycetophilidae and Cyaridae are the main pollinators, but these top 6 families, as well as other members of Nematosera as well. And here are order representations for each year's collection. In 2018, around 60% of the the specimens collected were of the order Diptera. Around 30% were Thysanoptera, and the other 10% was distributed among the other orders. Keep in mind that even though this category is listed as Diptera, the majority of Diptera collected were in the suborder Nematosera. 2019, there were 90% of specimens collected were of Diptera, and only 1% of specimens collected were Thysanoptera. And in 2020, the difference was even larger, with about 95% of specimens being Diptera and only 0.4% of specimens being Thysanoptera, with the other 5% being distributed among other orders. And for a cumulative graph, for the cumulative pie chart, the total for Diptera goes back down to 90% of specimens collected, while thrips finalizes at around 1% of specimens collected were Thysanoptera. With the only bigger category, with around 2.5% of specimens collected being Thysanoptera. With the only category being larger is Hemiptera at around 5%. Here's a graph comparing the specimens collected that were in the suborder Nematocera compared to those from Thysanoptera. And then finally, the gray bar just shows anything else that was not of Nematocera or Thysanoptera. And so Nematocera here there's so many more Nematocera specimens collected compared to Thysanoptera. And like I mentioned earlier, this shows at least some merit for Thysanoptera definitely not being the main pollinator of Jack-in-the-pulpit at Mount Briar, and gives more credit to the fact that Nematocera are the most likely to be pollinating Jack-in-the-pulpit, just due to the sheer numbers collected. Because even insects of all the other orders collected there were only 57 of them collected over the past 3 years. Well, over the past 3 years, there's 441 Nematocera. And in conclusion, a wide variety of insects visit Jack-in-the-pulpit as shown by my data. And Jack-in-the-pulpit could rely on an extremely complex system of this wide variety of insects for its growth and pollination, which could be why that its pollination proves to be a little bit ineffective. And there are also certain Nematocera families appear to be strongly associated with Jack-in-the-pulpit. And previous studies have stated that Cyarids and Mycetophilids were the main associations, but I think that, my data shows that it most likely extends outwards to the other families that were not mentioned previously, such as Psychotidae, Ceratopagonidae, and the other two. And with that information, Nematocera are potentially the main pollinators of Jack-in-the-pulpit, at least in Mount Briar. And if Nematocera nor thrips are pollinating the plant, then that could be what caused the evolution of cloning in the Jack-in-the-pulpit plants due to an ineffective pollination system. And finally, I would like to thank the Entomological Society of Washington for allowing me to share my findings here. I would like to also thank the Washington County Parks and Recreation for allowing me to collect at Mount Briar for the past 3 years. I would especially like to thank Dr. Adamski for supporting me and encouraging me over the past 3 years. I really do appreciate it. Dana DeRoche for identifying many of the spiders I collected to the family level. That information was not presented in this presentation, but I really do appreciate it. I'd also like to thank Dr. Gary Miller for identifying the aphids collected. And finally, I would like to thank Dr. Cheryl O'Donnell for identifying the specialist thrips, as well as preparing some very nice slides for me. And if anybody has any questions, I will try my best to answer them. And I would like to thank you all for listening.
That was great, Ian. Sorry, I'm just setting up my video now. Thank you so much. If anyone has any questions, please feel free to either drop your question in the chat or just speak up and unmute your mic.
Hi, Ian. This is Cheryl. I think you did a wonderful job. I really, truly appreciate the work that you put into this little research project. Actually, it's not that little. It's quite big. 3 years is a lot of data as well. So very nice. Nicely presented as well.
Thank you. I plan to actually collect even more data after this as well.
Oh, very good. Very good.
Ian, this is Claire Hudson. First of all, congratulations. What a fantastic presentation. You did awesome. I do have a couple of questions. So, you know how you were finding the spiders in the flowers? Was there did you notice any trends, whether they were in male or female flowers? I'm just curious because if you think about, you know, spiders being in the female flower, they might have more of an advantage if nothing escapes, right? Was there any sense of an observation there?
I didn't particularly pay attention, but I think it was pretty even among both sexes.
Mm-hmm.
Because
Cool.
generally, I think it can be assumed that if an insect went into the male plant spathe, it would take a little while before it found the exit opening.
Mm-hmm.
So that would probably give a spider enough time to catch it if it didn't have a web up. Or for the spiders that made a web inside the spathe, it probably didn't matter much if there was a hole or not, because I don't think anything flying in would be able to evade the amount of webbing that was inside.
Right, that's cool. And the other thing that occurred to me on your graph where you showed the distribution of your insects that you found in 2018, when it was rather wet that year, the thrips were higher. And I was wondering, does anybody know if they're particularly sensitive to rainfall, or does that have anything to do with it? Because I have no idea. It just popped out at me like, did that have anything to do with it? I don't know.
Well, as a Thysanoptera specialist, generally speaking, when you and there have been those that have broken the mold here, generally when you have a very wet year, there are a lower number of thrips. So that's a very interesting, you know, part of this study. I think it's very interesting.
Yeah, that is interesting. Very good. Well, excellent job, Ian. I'm so glad you sent me the link.
Yeah, great job, Ian. That was awesome. Thanks. I had a quick question just about the plant. You know, I don't think you would consider it like a carnivorous plant, but you mentioned that at least in the female plants, the some of the insects might fly in and get caught in there and not be able to fly out. Is there like any indication that the plant is somehow like deriving nutrients from the dead insects in there? And or has anybody suggested that something like that or broader in the arum family?
I'm not sure about the rest of the arum family, but I'm pretty sure that Jack-in-the-pulpit does not derive nutrients off of the dead insects in the bottom.
Okay.
I think it's more from a very inefficient pollination system, because females will have a lot more you'll find usually a lot more dead insects at the bottom due to the lack of an exit hole.
Okay. It's a cool plant.
It's a very interesting plant.
I know this wasn't necessarily what you were looking at, since you were mostly looking at the upper part of the plant, but did you ever notice insects or arthropods around the roots of the plant, like decomposers?
As decomposers, I did not observe any. However, there were those aphids found at the base of the stem, underground actually. I'm not entirely sure what their association is with the plant. But as far as anything eating the plant, I'm not aware of anything that feeds on it or uses it as a host plant besides the specialist thrips.
Any more questions? Okay, well, I will pass it off to Cheryl. I think Gary Hevel is on the line to read our minutes, hopefully.
Thank you, Cece. Gary, are you available?
Yes, we just got on.
Okay, super. Gary, can you read the minutes from our last meeting?
Yes, we'll try.
Okay.
Can you hear me well enough to broadcast it, or?
Yes.
Okay, good. The 1214th regular meeting of the Entomological Society of Washington was convened at 7 p.m. on the 1st of October 2020, and for the first time in the history of the Society, a virtual meeting facilitated by a Zoom application. A total of 46 people was registered, including both guests and visitors. Society President Cheryl O'Connell asked Recording Secretary Gary Hebel to read the minutes of the March meeting. Gary replied that the notes he took in March are at the National Museum of Natural History, which has been closed since mid-March and thus not available at this time. Gary promised a later reading of the referenced meetings minutes following the opening of the museum. Cheryl then asked Membership and Communications Secretary Christina Say for information on new members of the Society. Christina responded that she had not assembled a list of new members and would provide this information at the next meeting. Cheryl noted that the Executive Board of the Society has been meeting through the summer, mostly toward the goal of a new website for the Society. David Adamski mentioned that the Young Entomology Group met in February and March and hoped to have a virtual meeting in December. Program Chair Cecilia Escobar noted that she will not continue in that position for next year and asked members for suggestions of replacement candidates. Relative to the program of the evening, Stuart McCamey offered information that the O'Brien collection was almost donated to the Smithsonian, but a number of factors led to it going to Arizona State University. Program Chair Cecilia Escobar introduced the evening presentation, a 34-minute documentary film, The Love Bugs. Since its release in 2018, this film has been viewed many times, especially at entomology societies, including the Maryland Entomological Society. The subjects of the film are Charlie and Lois O'Brien, renowned entomologists who have resided in Green Valley, Arizona for many years. The film focuses on the donation of the O'Brien collection to Arizona State University and the preparation of the collection for transport to ASU. Many students were involved in the transfer, all of whom were thrilled to participate in the lengthy procedure. Throughout the film, Lois and Charlie are shown as loving partners, respectful of each other, but quibbling over details in their conversations. Charlie notes that he is 84 years old and has suffered from Parkinson's disease for the past 25 years. He regrets not being strong enough to lift and carry insect drawers, leaving this task to Lois. He has fallen 21 times in the past 15 months. Lois acts as his caretaker. Charlie is boldly brave about his condition and prognosis and states that I'm going to be cremated because I don't give a damn about this other stuff. He and Lois are widely known as major collectors and scientific contributors. He has studied weevils while Lois works on fulguroids. They have traveled to 67 countries and territories of the world with the purpose of collecting insects. These efforts have resulted in the accumulation of more than 1 million insects that fill 1,268 insect drawers valued at $10 million. At ASU, a database will record the specimens with a concentration of species from Mexico. Warren Steiner commented that in 2013, because of a rainstorm during a collecting event, he spent the night in Green Valley on a couch in the O'Brien house. The meeting was officially adjourned at 7:50 p.m.
Thank you very much, Gary. I will ask the members if anyone has any objections or corrections to the minutes.
I'd like the motion that the minutes be accepted as read.
Thank you very much for the
Second.
motion. Second?
Second.
Thank you. Then we will accept those minutes as read. Thank you very much, Gary. It's greatly appreciated.
Sure.
All right. I want to say thank you to our presenter, Ian. That was a wonderful presentation. It was lovely to have a young entomologist come in and provide us with such a wonderful presentation. And I look forward to our next meeting, which will be December. I just had it in my head. Anybody else think about it? It's December. Hold on.
Third.
3rd. Thank you. I was going to say that and I thought, no. So do we have any other business that needs to be presented here tonight? All right. Do you have a motion for adjournment?
Motion.
Second?
Second.
All right, everyone. Thank you so very much, and we will talk to you next month.
Thank you for joining.
Thank you.
Cheryl. Thank you, Cece.
Thank you very much, guys.
Thank you for having me.
Oh, you bet. That was great.